Okay, so the last talk for this session is from Peter Nguyen from Cedars-Sinai Medical Center, that is going to talk about a molecular taxonomy of tumors independent of tissue of origin.
Okay, thank you. Hello, my name is Peter Nguyen, and I'm a third-year PhD student in the laboratory of Dennis Hazlett at Cedars-Sinai Medical Center in Los Angeles, California. And today I will be presenting the research our lab has done entitled A Molecular Taxonomy of Tumors Independent of Tissue of Origin, where we identify pathway disrupted due to gene disruption, and we attempt to reclassify these tumors based on pathway disruption. So cancers are typically classified using pathological criteria that heavily rely on tissue of origin. And this traditional method of classification paves the possible treatments route for an individual with a specific cancer type. Yet two persistent observations that frustrate the primacy of tissue of origin in cancer are: one, there are few drugs or treatment that can be said to be largely curative for a specific cancer type, and two, some drugs work against multiple cancers. These observations suggest that tissue of origin fails to serve as an adequate proxy for some fundamental molecular mechanism driving responsiveness to treatment. So if you go back 20 years ago, the idea that all of the genes altered in cancer could be identified at base pair resolution would have seemed like science fiction. Today, such genome-wide analysis through sequencing of whole exome or genome is routine. As a result, data sets have become available that would allow researchers to understand the genomic landscape of cancer. For example, the Cancer Genome Atlas has molecularly characterized 33 different tumor types from a cohort of 11,000 patients. And these data types include mutation, copy number, gene expression, methylation, and so forth. And while the data provides a comprehensive view of an individual tumor, the complexity in interpreting the data remains a challenge. Considerable progress has been made analyzing these data. Statistical analysis identify hundreds of global and tissue-specific cancer driver genes. And more recently, pan-cancer mutations and other data have been looked at as disruption of normal pathway activity. Other studies provided a comprehensive view of mutation, gene expression, and genomic signatures. Hadley's motivation was to reclassify tumors using molecular features. In 2014, they used TCGA's first data release comprising of 12 different tumor types and applied their cluster of cluster algorithm. Their molecular clustering resulted in 13 clusters dominated by tissue of origin, due to the fact that most of the clusters were a near one-to-one relationship with tissue of origin. In 2018, they revisited this, and given that their previous analysis only included a third of the final set of TCGA samples. Again, they found a similar theme: tissue of origin dominates molecular clustering. So the big question is whether tissue of origin can be split into molecular subtypes. Because cancer is a genomic disease, we need to transform our understanding of cancer by moving away from this overly simplified traditional model for cancer therapy, a one-size-fits-all, to a precision and personalized medicine approach. And this will greatly change clinical practice, drug development, and the way we diagnose and treat a wide range of cancers. Our hypothesis is that cancer represents several unique combinations of molecular dysfunction in basic universal cellular processes that are both viable and confer selective advantage. So our general approach to testing our hypothesis is to acquire whole exome sequencing data from TCGA because the affected target genes is known. And below is a bar plot that shows the number of samples for each cancer type. To study molecular dysfunction, we focused our analysis using deleterious somatic mutations, such as a stop gain and frame shift mutation. And finally, in order to strip away tissue of origin signatures, we capture molecular dysfunction by converting genes into pathways. So we first started off with our gene mutation data, which consists of the gene mutation status for 7,600 tumor samples. We excluded genes with low or no expression in each tissue type. The way we did this is setting a threshold value, and genes that did not meet this threshold value were not considered expressed in our analysis. We used Reactome, which is a knowledge-based pathway database, to map genes to pathways. We imposed several criteria in selecting our pathways. It had to be human-derived, a molecular pathway, and it had to be non-disease related. And so using this lookup table, we modified the data from a gene-centric to a pathway-centric. And additionally, we were not interested in how many times the pathway was mutated. We were only interested in if the pathway was mutated or not. And this type of transformation removes any bias that would have been seen due to mutation rate. And our in silico analysis showed that mutation rate and the number of genes mutated were positively correlated. And after all these observations, this is how a small subset of the data looks like on the heat map, where the rows are the 377 reactome pathways and the columns are the tumor samples sorted by their cancer type. And red means the pathway is mutated and gray means the pathway is not mutated. And what we observe is the mutation profiles were not tissue specific. So we decided to classify our tumors based on pathway disruption. We used multiple correspondence analysis, a technique for analyzing categorical variables, to analyze patterns of pathway mutation relationships between samples. And to summarize the results graphically, we use UMAP. So here is a UMAP of that operation in 3 dimensions. We use hierarchical density-based clustering to identify high confidence density clusters and observe 10 distinguishable clusters. So this new updated UMAP has tumor samples annotated by their cluster identity, and tumor samples that were not assigned to a cluster were colored gray, as shown here. And finally, we use K nearest neighbor to assign the unclustered tumors to their closest cluster. So to visualize the distribution of cancer types within the cluster, we generated a Sankey plot where the left node represents the cluster and the right represents cancer type, and the flow shows the relationship between them. So for example, cluster 10 here, you have some uterine, some stomach, some lung, and so forth. And so to reinforce this, we projected several cancer types onto the UMAP, and most cancers are well distributed across the clusters. This suggests that our pathway-based clustering is independent of tissue of origin. Next, we looked at other variables that could potentially drive the clusters. The four major subtypes of breast cancer correspond to histological and molecular expression profiles that define them. And consistent with this, we found that breast tumor samples of luminal A subtypes, shown here in light blue, were biased towards clusters 1 and 7. And basal-like colored in yellow was biased towards cluster 6. However, both subtypes can be found in every other cluster, and luminal B and HER2 positive were found across the clusters, indicating no cluster exclusivity. Similar to breast cancer, we also observed a similar theme for head, neck, and liver, as indicated by a mixture of colors. We projected somatic mutations in genes involved in genome instability, such as BRCA1 and 2 mutation, and did not see any obvious trend. We projected metastasis status and found that metastatic tumors colored in dark blue were distributed across the cluster. We projected tumor stage and found a similar results. We also projected patient attributes such as age of diagnosis and gender and found the same thing. And finally, we looked at mutation rate of a cluster, and apart from 1 to 2 clusters, mutation rate is not a driver of cluster identity. So next, we asked the question, what's different about them? And one way to do this is to find enrichment of a pathway disruption in a cluster relative to the background using the beta distribution. So for example, if we're interested in cluster 2, we would estimate the mutation rate for a cluster based on the observed and compare it to the background outlined in blue. And if the mutation rate of the cluster is greater than the background, then we say that's enriched. But if it's less than the background, we say it's depleted. So the heat map on the right here shows the effect size of each pathway for each cluster, where orange indicates pathway mutation enrichment and purple indicates depletion. And we observed distinct mutational pathway enrichments between each cluster, but we also noticed some shared pathways, such as in clusters 7 through 9 here. And this is consistent with the UMAP where you can see cluster 7, 8, and 9. So to further investigate the enriched pathways, we filtered for pathways with an effect size greater than 0.3. And clusters 7 through 9 were more strongly defined by disruption in extracellular and intracellular signaling, whereas 3, 5, and 6 were found to have disruption in metabolic pathways. Cluster 4 had disruption in regulation of DNA and histone methylation. And cluster 2 had disruption in a various amount of pathways related to cancer. So up until now, what we presented is that our pathway level classification is independent of tissue of origin. subtype, mutation rate, and so forth, and that these clusters represent distinct pathway disruption. So this raises the question, is our pathway-based clustering driven by a few set of common driver genes? To do this, we compare the top pathway genes for each cluster relative to the TCGA background to find differentially mutated genes. We ranked the log odds ratio and selected the top 10 enriched and depleted genes with a p-value of less than 0.01 for each cluster. And on the right of that, on the right is the result of that operation, where genes that are to the left of this black dotted line are depleted, where genes to the right of this is enriched. And the plots are colored by their cluster identity. So you have cluster 1 and 2, 3, and 4. So cluster 7 and 8 right here, which shared multiple enrichment and signaling pathway, have an overabundance of mutation in PI3K and RAS mutations, respectively. Cluster 2 had mutations in proteasomal subunits involved in protein degradation. Cluster 4 had mutations in the nucleosome structure. And we also observed genes that were enriched for one cluster are depleted from others. So for example, we observed a depletion of PI3K mutation in clusters 3 and 8, but we saw an enrichment in cluster 7 and 9. So these are the same odds ratio plots, but from the previous slide, but what I've added to this is a heat map that shows the proportion of samples with a mutation in each gene broken down by tissue and class combination. So with the exception of P53 and PI3K, clusters were not predominated by one or more highly mutated genes across all cancers. So for example here, in cluster 4, when observing the mutation rate for these top enriched genes, the mutation rate is heterogeneous. An easy observation is the gene mutation pattern for cervical, glioblastoma, and skin. It reveals how tissue-specific signatures can have similar pathway disruption. And even when comparing the PI3K mutation, where you can see I've outlined it green here for clusters 3 and 8, which have a depletion in PI3K mutation, and 7 and 9, which have an enrichment. When comparing within the two groups or all of the groups, they do not show a similar trend. So there's no global pattern indicating that our clusters are not driven by individual genes, but rather networks as a whole. So if our clusters represent biological states that are distinct from tissue of origin, they may have different prognosis within cancer type or across all cancers. To illustrate this, is a cancer from cluster 2 more deadly than one from cluster 6? So to explore this idea, we use Bayesian inference to test models of survival using the public longevity data from the CDC. And because most cancers are diagnosed later in life, we fit a model that emphasizes an accuracy on the right tail of the distribution, as shown in the Kaplan-Meier plot on the right, which compares the actual CDC data with the model fit for men and women. So we model baseline longevity using a risk rate function. The model assumes that the risk of death at age 20 years old increases exponentially with a constant rate all the way up to your age of diagnosis. And then risk of death is assumed to have a new rate thereafter when you're diagnosed with cancer. Also, we found that it was not necessary to use the actual age, but instead an effective age for a given tissue class combination. Our effective age is calculated as follows. A tissue times A class determines the effective age, and the effective age increases by some amount based on your actual age. So basically what all of this is saying is if you're a 50-year-old diagnosed with stomach cancer, you basically now have an average survival rate similar to that of a healthy 82-year-old based on the posterior probability of stomach cancer. And lastly, we have K tissue, which dictates how fast you age in a normal year. And the red dotted line here indicate the values for a non-cancer patient. And to further illustrate the model, we simulated survival for 4 related groups. We started off with a cohort of women age 30 and show their expected survival in light blue. And now comparing this to randomly selected women ranging from 30 to 70-year-old in green, you can see that the survival changes due to a mixture of age. And then here we show the survival rate for a 30-year-old with breast cancer, which further reduces the expected lifespan. And of course, mixture of age reduces it even further. So our model estimates survival without confounding to the effect of age. And finally, here we show an example of how we applied the cluster-specific aging multiplier for ovarian cancer to investigate whether if we observe any differences in survival between the clusters. So if you focus your attention here to the posterior probability distribution for ovarian cancer, which is sorted by their cancer cluster identity, the red dotted line here indicates the aging rate for an average ovarian cancers. So tumor classes like 4 and 9 are less deadly compared to the rest of the cluster, which is shown here on the survival plot on the right, where you have 4 and 9 here being better. So to summarize, we sought to simplify the problem of tissue of origin by employing a knowledge-based driven analysis. We accomplished this by using a pathway-centric approach. We find that this approach is independent of tissue of origin, among other things, and we show that these clusters have differences in pathway enrichment. And by modeling the CDC longevity data, we were able to show the effects of cancer and cluster membership on survival expectancy. And if you would like to learn more about the results from our paper, you can visit our interactive media to explore the clusters as well as simulate survival for your favorite cancer type and class combination. So here in the pan-cancer tab, you can explore the UMAP structure with the ability to project different variables. The UMAP structure is pan and zoomed enable. The cancer type tab will allow you to look at the distribution of your favorite cancer type and their subtypes on the UMAP. You also have the ability to select more than one cancer type. In the gene tab, you can visualize the distribution of your cancer and your gene mutation on a UMAP. And of course, you have the ability to choose more than one cancer type. And the pathway tab is the same as the gene tab, and now you're looking at pathway disruption. And lastly, the survival tab allows you to simulate survival based on cancer type, cluster identity, sex, and age. And here's a short video of how you create something that I showed on my previous slide. To generate a 30-year-old with breast cancer, you would adjust the age range to only include 30-year-old. You click show control, which will give you the survival curve for a non-cancer patient. And you can adjust the population size to have more samples. And finally, you can also simulate survival based on cluster identity. And so this would have not been possible without the tremendous help from Dennis, Simon, and Daniel. And special thanks to these 6 individuals here for their feedback and comments to the project and the funding that supported this project. And with that, if you have any questions, please let me know. Thank you.
Thank you very much, Peter. Okay, so I see no questions at the moment, but oh no, one just popped out. So have you examined the pathway clusters with patient response to targeted therapies correlating with different pathways?
Currently, right now, the way TCGA the TCGA databases has, it's not very easy to interpret that. And what we've thought about doing is using cell line data, where we can recluster we can assign cluster membership to the cell lines, and then there's pharmacogenomic studies done on these cell lines, but we haven't done that yet, and that's one of our future projects that we want to do.
Thank you. I was curious about whether you explored maybe to use different clustering algorithms, or how the number of clusters that you found was set. So if you explored like a different number of clusters or a different type of clustering method, and if the results confirmed what you obtained.
So the way we defined our cluster was we use HTTP scan, which has one hyperparameter, which is the minimum cluster size. And what we did was we optimized HDB scans so that it would have the fewest number of unclustered samples. And we do kind of like iterations of 1,000 times to find, or 100 times, sorry, 100 times to find the most, the cluster, minimum cluster size that has the lowest amount of unclustered samples. And HDB scan determines how many, you don't need to tell HDB scan how many clusters you want. It will determine that for you. Yeah.
Clear, thank you very much. Okay, so I see no questions left. So I guess we can conclude